Okay, now we go to the next part of the workshop. Now we'll talk about the early genetic markers we use for genetic mapping. Okay, actually, genes were the first markers to be used for the genetic mapping. Okay, in the early genetic maps, the Alfred Sturtevant created the first genetic map of the chromosome Drosophila melancestor in 1913. Okay, genes are arranged on the chromosome in a linear way. Okay, genes are present on the chromosomes in a linear way. Okay, the first thing. The next thing is the genes for specific traits are located in a particular places. Okay, if the genes are present here, it's always present here. Okay, it will not shuffle into the next part that is very close to each other. Okay, it will shuffle to the very far but not in the near locations. Okay, frequency of the crossing over between the two genes could help determine their location of the chromosomes. Okay, if the two genes are crossover between it, so by the this frequency between the genes actually help us to determine the location of the chromosomes. Okay, genes that were far apart, okay, on the chromosomes are more likely to inherit it separately. It means they are not inherited together. So by the genes we can locate the genes very easily. Like things, there are the pair of the chromosomes, okay, that are homologous to each other. Chromosomes crossover, recombination. If there is a, okay, there is a crossing over is actually occurring, there is a crossover done. Okay, now you see the genes are shuffled, recombinant chromosomes are there. So the genes that are close to each other on the chromosomes are more likely to inherit together. Okay, it means that two genes that are very close to each other. Sorry. Uh, okay, I talk about the early genetic markers. Okay, early genetic markers in which the genes were the first markers to be used for genetic mapping. Okay, actually genes are arranged on the chromosomes in a linear way. Okay, genes are present on the chromosomes in the linear way, and genes for specific traits are located in a particular places. Okay, and by the genes for the specific traits are located in a particular places. Okay, and the frequency between the crossing over between the two genes actually determine us the location chromosomes, the location on the chromosomes. Okay, the genes are where the genes actually the located, and genes that were far apart on the chromosomes are more likely to be inherited separately. And like this, these two genes are very close to each other. Okay, it means they are not separated, they are not shuffled. Okay, in that part there are no shuffle, but the distance between the B and C is very high. Okay, it means they are shuffled. Like this, they are shuffled. It means if there is a high distance between the two genes, the recombination frequency is very high. Okay, I'm going to check it also. Okay, if there's small screen, so you please off the captions. If you off the captions, you can see the large screen also, or type on the mobile to make it big, because in my side it's okay like this. Okay, now we talk about the genetic map of the chromosomes from the fruit fly. It's actually in the fruit fly, the chromosome, like the yellow body, the white eyes, the wings, the bristles, the tan body, the miniature wings, the sable body. Those are all present in there. Okay, and there is a recombination frequencies between the them. Okay, it means there is a very little difference distance between the two genes. There is just a 1.5 recombination frequency, and about 1.5 to 13.7 there is a long distance between them. Okay, now we talk about the modern genetic markers. Okay, in which in which actually we talk about the advantages and the disadvantages of the genes. Then we talk about the modern genetic maps. Genes are very useful markers, but they are by no means ideal. It's not a ideal, it's not a perfect. The advantages of the gene markers is genes are directly associated with the phenotypes. Okay, genes are close to each other. Okay, there is a okay after 27 minutes. Okay, we talk about this. Actually, the advantages of the genes is that genes are directly associated with the phenotypes and biological functions. Okay, if there is a gene, it's actually transcribed into RNA. Okay, and that RNA is translated into proteins. Okay, and that proteins actually make us. Okay, the make the proteins actually make the cells, make our body. It means there is if there is any problem in the gene, it means the protein is also affected, and if the protein is affected, our phenotype, our the behavior, our character will also be affected. Okay, and it means genes are directly associated with the phenotypes. Okay, and by checking, by analyzing the phenotype, we can check the genetic mapping, like where the genes are actually located. Okay, the inheritance of the genes can be easily correlated with the observable traits, aiding in the linkage analysis. Okay, if the genes are by checking the linkage analysis. Okay, if the crossing over is very high, it means the genes are located very far to each other and it's not inherited together. Genes are essential for identifying loci associated with the hereditary diseases. It's like saying if the protein is affected, we will get the disease. Okay, and by reverse check it, we can check the genes. Okay, and we check the genes expression. If the genes expression is off, we can check, we can infer it there is a problem in the there is a problem in the gene, and by this we can identify the location of the genes. And there is the limitations of the genes. That is why we have to go to the modern genetic markers. Actually, there is a number of identified genes may be small. Okay, there is a, almost the 20,000 genes are present on the chromosomes. Okay, but only the 50% genes we know. Actually, we have information about the genes that what works they actually do. Okay, that is why the number of identified genes may be small, restricting their utility as markers. Okay, genes are not evenly distributed along the chromosomes. Okay, not evenly distributed. Some genes are very far, very close to each other. Some genes are very far from each other. Okay, so there is a hotspot of the genes where the genes are present in very high number, but where the genes, some points where the genes are not, rarely present, actually the very rarely present. Okay, recombination hotspots, there is the regions with high recombination rates. Okay, there is the recombination is very low. Okay, because the crossing over between the genes is very low. The genes are always inherited together, but the combination is very high. It means the hotspot, it is the hotspot region for the recombination. It But these hotspot regions actually disrupt the linkage relationships. Many traits are polygenic. Means that one phenotype or the one trait is actually controlled by different genes. Okay, one phenotype is actually controlled by different genes. So by this we cannot identify, we cannot determine actually the location of the gene by checking the phenotype of the trait of producing by the single gene, because one single gene is actually not present in the trait. Using the single gene as a markers may not be provided the enough resolutions. That is why a map based on genes are not very comprehensive. Okay, that is why we have to go to the we have to go to the modern genetic markers. Okay, there is any question about the genes, the genetic mapping till now, can question me in the chat box or by opening the mic. Yeah. Okay, you can also stop me if you have any question. You can stop me and ask me the question, and then we have to further go to the workshop. Now we talk about actually the modern genetic maps. Okay, the DNA markers were used for mapping. Okay, and the DNA marker has at least two alleles to be useful. Okay, so there is a in the DNA markers there is a two alleles to be useful. Okay, it means in the one, and so there is a second homologous chromosomes. Okay, and there is a one gene is present here and the second gene is present here. It means the two alleles are present here. Okay, so there is just a two alleles that will be very useful. The three types of DNA sequences features that satisfy these requirements. Okay, first of all is the RFLPs, the second one is the SSLPs, and the third one is the SNPs. We first of all talk about the RFLPs. Actually, RFLPs, it is the acronym of restriction fragment length polymorphisms. Okay, in which it's actually the first type of DNA marker to be studied. It refers to the differences among the people in their DNA sequences that the sites recognized by the restriction enzymes. Actually, for example, it's a it's a long DNA. Okay, and in there is a restriction sites. Okay, restriction sites is actually the special sequences at which the restriction enzymes cut. The restriction enzymes cut, there is a different types of restriction enzymes that cut the different types of the sequences. For example, the one restriction enzyme cut at that point, that point, that point, that point. If there is any mutation in the restriction site, the restriction enzyme will not cut that site. Okay, and by this we can know about the we can know about where the genes are actually located. Okay, it refers to the differences among the people in their DNA sequences at the size recognized by the restriction enzymes. Such variations result in different sized DNA fragments produced by the digesting the DNA with the restriction enzymes. Okay, by this we get the variations. Okay, some DNA will be cut by this, some DNA is not cut by the restriction enzymes. Okay, by this we get the different types of, one DNA fragment is there, one DNA is very small, one DNA is very large. Okay, so we get the different types of DNA fragments. Okay, RFLPs can be used as genetic markers which are often used to follow the inheritance of the DNA through the families. Okay, it means if the genes, if the restriction enzymes are very close to each other, okay, it means they are not inherited together. If the restriction enzymes are very far from each other, it means it's not always inherited together. Okay, treatment of DNA molecule with the restriction enzyme produces the same set of fragments. Okay, if we if we treat the DNA with the restriction enzyme, the same restriction enzyme, it produces the same set of fragments. Okay, same set of DNA. It means the same size of the fragments will be get by the same restriction enzyme. If there is a no mutation, okay, if there is a no mutation, the same size of the fragments we will get. With the genomic DNA, some restriction sites are polymorphic. Okay, with the genomic DNA, some restriction sites are polymorphic. Oh, what is polymorphic? It means the polymorphism, because individual to individual there is a different types of mutations in our DNA. Okay, if the mutations are present in the restriction site, okay, if the restriction if the mutation is present in the restriction site, the restriction enzyme will not cut it. If the restriction enzyme will not cut it, the fragment length is very long. Okay, and by gel we can identify the differences between the fragments, and by this we can identify, we can analyze it, and we can check the polymorphism between the different individuals. Okay, the sequence alterations result in two adjacent restriction fragments remain linked together. Okay, if there is a alteration, if there is a mutation, it means the two restriction fragments will be remained linked together after treatment with the enzyme, leading to the length polymorphism. Okay, we'll talk about the first. If there is a mutation, restriction site will be mutated. If the restriction site will be mutated, restriction enzyme will not digest it. If restriction enzyme will not digest it, it means, okay, like this. It's a DNA. Okay, and we add the restriction endonucleases or the restriction enzymes here. And if there is a four fragments, okay, one fragment is cut here, one cut is here, and one cut is here. By this the four fragments will be produced. But if there is a mutation between them, okay, if there is a mutation is here, it means that there is a three fragments will be produced. Okay, okay, now we'll talk about the example. For example, there is a mapping a gene for disease resistance in the plants. Okay, if there are the plants that are resistant to the disease. Okay, and now we have to mapping the gene that is actually resist that plants from the disease. A scientist want to find a gene in the rice that gives resistance to a specific disease. First of all, they choose the population. Okay, first of all they choose the population. They collect the DNA from a group of rice plants. Some resistant to the disease, some are not. Okay, it means that some are resistant to some disease and some are resistant to not to the disease. Now do you hear me? Okay, sorry. Ajay, now do you hear me? First of all, if a scientists want to find a gene in the rice that gives the resistance to a specific disease, first of all they have to choose a population. Okay, and they collect the DNA from a group of rice plants. Some are resistant to the disease and some are not. Okay, they cut the DNA. Okay, now they cut the DNA and make the fragments by restriction enzymes. They cut the DNA by restriction enzymes, producing the fragments of different sizes depending on the genetic sequence. Compare the fragment patterns. The fragments are separated on a gel and compared. Okay, and resistant plants might have a specific fragment that are non-resistant plants lack. Okay, it means the resistant plants might have specific fragments. There is a mutation in the resistant plants that might have different types of the fragments that are not present in the non-resistant plants, and identify the linked marker. Okay, by this we check where is actually not cut, where is the point where the fragment is not cut. We identify that point. Okay, we identify that point and the position of the marker in the genomic mapping. And by analyzing the inheritance pattern, we check the inheritance pattern. It means if they are inherited together, it means these genes are very close to each other, and by this we locate the genes. Scientists estimate how far these markers is from the known markers. Okay, and by placing it on the genetic map, and by this we can locate the genes. Okay, now we will talk about the SSLPs. In the simple sequence length polymorphism SSLPs, we actually check the variations resulting in the leaves of the different lengths. Okay, in our DNA, there are different types of repeats are present. Okay, there is a STRs, there are VNTRs, there are microsatellites, there are macrosatellites, there are mini satellites are present in our DNA. Okay, the the variations between the individuals. Okay, the differences between the individuals by this repeats difference, by this the number of differences, number of repeats. Okay, by the differences on the number of repeats, we can identify, we can determine the genes location on the chromosomes. Okay, SSLP is a type of genetic variation. It's actually a genetic variation resulting in the alleles of the different lengths that are present in the different individuals. SSLPs are arrays of the repeat sequences that display length variations, okay, in the different alleles containing the different number of repeats. Okay, now two variants of the SSLP. Okay, first of all, this is the allele. Okay, in which there are just three repeats. Okay, but in the allele 2 there is a 2, 3, 4, and there is a 5 repeats. Okay, there is a 5 repeats and there are just 3 repeats. Okay, it means there is a variation between the two alleles. Okay, and it's also a difference between the, if it is present in the one individual and another individual in which there is a difference is just 3 and 5. Okay, it's also heterozygous, means these two individuals are related to each other. They may be a relatives, there may be a mom and son, or they are parents of the one offspring. It means that. Okay, the RFLPs are biallelic, while the SLPs can be multiallelic. Okay, why? In the RFLPs, there is some mutation, okay, or there is no mutation. Okay, there is there is a mutation or there is no mutation. If there is a mutation, it means the cut will be occur. If there is, okay, if there is a mutation, may or not the cut will be occur. If there is no mutation, the cut may or may not will occur. Okay, there is just two positions, or there is just a two circumstances. The cut will be occurred or cut not will be occurred. It means in the RFLP there is just a two allelic, there is just a biallelic positions. But in the SSLPs there is a multi-allelic. It means that in the one person the repeats are five times. Okay, the microsite repeats are five times, but in the person two the repeats are six times, and the person three the repeats are three times. In the person four the repeats may be a four times. It means it's a multi-allelic. Okay, there is a no specific alleles between the SSPs. The variations are multi-allelic. In contrast, in the RFLP marker, it would two options: DNA fragment cut by the section enzymes, or the DNA fragment is not cut by the enzymes. Okay, there is a concept you have to know about it. The RFLPs are biallelic, but the SSLPs are multi-allelic. Okay, how SLPs are actually typed? Now we talk about the principle of the SLPs, and in the next slide we will talk about the workflow of SLPs. In the SSLPs, the analyzed using the PCR. Okay, we analyze it by using the PCR, in which based on the combinations of the SLPs and the PCR, the types are identified through the length of the amplified DNA fragments. Actually, we have, for example, we have a this type of DNA, okay, in which the repeats are present, and this DNA in which the repeats are present. Okay, we actually design the primers for them. Okay, the forward primers and the reverse prime, and we amplify these sites. Okay, and by PCR amplifies the DNA region containing the SLP and the length variation. The amplified fragments vary in length. Okay, the first have this fragment in which the five times repeats are present, and in which three times repeats are present. Okay, it means that there is a variations, and we visualize it by using the gel electrophoresis. Okay, now there is the steps. First of all, we have to design the primers by in silico. Okay, in the first workshop of the genetics and molecular biology workflow, we talk about the design primers, how to design the primers. We share the link of the recorded version of the workshop to you, in which you can also check how the primers are actually designed. We design the primers by